In this video, we'll discuss pyruvate oxidation and the citric acid cycle. All right, this fits into our larger discussion of cellular respiration, which is diagrammed here. Recall that at the end of glycolysis, glucose has been broken down into two molecules of pyruvate with 3 carbons each. And glycolysis is an anaerobic process, meaning that no oxygen is required. Now, if oxygen is present, the remaining steps of aerobic cellular respiration can take place, including pyruvate oxidation and the citric acid cycle. This is going to start with pyruvate being transported into the matrix of one of the cell's many mitochondria. Recall that glycolysis takes place in the cytoplasm, which you see at the top of this diagram. Glucose is transported into the cell. Over the course of glycolysis, it's broken into 2 molecules of pyruvate. And once again, if oxygen is present, pyruvate will be transported into the matrix of the mitochondria. And the matrix is where pyruvate oxidation into acetyl-CoA will take place, followed by the citric acid cycle. Okay, so we've transported pyruvate into the mitochondrial matrix, and now pyruvate will be oxidized, meaning that electrons will be removed. Where will these electrons go? Well, if you guessed that they would be accepted by NAD+, yay, well done. NAD+ will accept the electrons, thereby being reduced to NADH. In the process of oxidation, each pyruvate, which is a 3-carbon molecule, will be converted into an acetyl group, which has 2 carbon atoms. Okay, so where does the additional carbon go? It is bound to 2 oxygen atoms and released as a molecule of carbon dioxide. All right, and where does carbon dioxide go? Well, we're done with it in terms of cellular respiration. It's going to diffuse out of the cell. And in humans and other animals, it's going to make its way into the bloodstream, the lungs, and then eventually be exhaled. Okay, so our acetyl group, which is attached to a carrier molecule called coenzyme A, or abbreviated CoA, then enters the citric acid cycle. The citric acid cycle, which is also sometimes called the Krebs cycle or the TCA cycle, will complete the catabolism or breakdown of the original molecule of glucose. Like glycolysis, the citric acid cycle is a series of reactions, each catalyzed by a specific enzyme. Unlike glycolysis, however, the citric acid cycle starts and ends with the same compound, oxaloacetate. This is a 4-carbon compound which is regenerated after the electrons are harvested from the acetyl group, which is fed into it from pyruvate oxidation. We start with the acetyl group being transferred from CoA and added to that 4-carbon compound, oxaloacetate, to produce a 6-carbon compound, which is citrate. A series of oxidation reactions then harvest the high-energy electrons. In the process, 2 carbon dioxide molecules are released per acetyl group. All right, so these represent the last carbons that were remaining from our original glucose molecule. Remember, the citric acid cycle is going to go around once for each pyruvate, and we had 2 pyruvates for our original starting glucose. So a total of 4 carbon dioxide molecules will be produced, and this will complete our catabolism of glucose in terms of the carbon. All right, the remaining 4 carbons will be rearranged to that starting compound, oxaloacetate, which will now be ready to be bound to another acetyl group. And you can see how it is therefore a cycle. The citric acid cycle results in 3 NAD+ molecules being reduced to NADH as they accept high-energy electrons, and 1 FAD molecule being reduced to FADH2. Recall that NADH is going to deliver those high-energy electrons to complex 1 of the electron transport chain, and FADH2 will deliver its electrons to complex 2 of the electron transport chain. In addition, an ATP or GTP molecule, depending on the type of cell in which the citric acid cycle takes place, is produced via substrate-level phosphorylation. All right, so by the end of the citric acid cycle, all of the carbon that was present in the original molecule of glucose will be present in molecules of carbon dioxide. All right, but remember, the point of the whole process isn't really to break down glucose per se. It's to harvest electrons from glucose in our first three steps, glycolysis, pyruvate oxidation, and the citric acid cycle, that can then be used to power the electron transport chain, which allows chemiosmosis and therefore the bulk of ATP production within the cell.